We will be going over our article presentation, which is based on molecular evolution of chloroplast DNA sequences. This article is important because it helps scientists learn about the genetics of traits like flower color and how these traits can change. The research focused on Ipomoea or morning glory flowers because it is easy to access flower color, and flower color is important for pollinator attraction in some species. The authors Michael T. Clegg and Mary L. Durbin are plant geneticists at the University of California, Irvine. We chose this article because we want to learn about plant genetics in an environment and how the environment can affect flower color in different ways.
Methodology 2.1 Sampling, PCR, and Sequencing. In the article, researchers tested 90 Puma species based on how their flowers displayed color, also known as anthocyanin and pigment. They also used two Ipomoea plants. One of them was normal and the other one was mutant. These two were used to study pigment gene regulation. To test out the experiment, flowers were put into greenhouse conditions. Researchers collected parts such as limb, tube, anthers, and stigma from open flowers. These parts were frozen into liquid nitrogen for RNA extraction. After RNA had been extracted from pigment tissues, but for the flowers that had no color, which were I-alpha RNA, was taken from another colored part known as a hypothosal. cDNA was treated with DNase, primers developed from known Ipomoea genes. CHSD, CHSE, CHI, DFRB were utilized to amplify each gene in all species. PCR products were then sequenced using Big Dye Terminator chemistry on an ABI 377 sequencer. Direct sequencing would be one of the options, but if failed, the products would be cloned, then replicated and sequenced at least 8 clones to be sure to avoid PCR errors. Methodology 2.2 and 2.3. The sequence were then arranged in conscious using Fred, Frapp, Conced, FollyFred software. This arrangement of the 4 genes was uncomplicated. Phylogenetic trees was found formed under maximum likelihood analysis in PAUP. The total RNA was then extracted using a method of O'Neill et al. 15 micrograms of RNA were also used to run 1.2% agarose formaldehyde gels, which then were transferred to green screen filters. Blots were UV cross-linked and assayed for the amount loaded with RNA bands. They were washed under high stringency to minimize cross-hybridization. Probes were specific to the different portions of the genes CHSD, CHSE, DFRB, and CHI. Methodology 2.4. Lastly, the flower tissues were fixed, embedded in paraffin, and then cut into 10 microgram sections. The Digsazation labeled RNA probe system was used to cut the sections. SHSD and DFRB were labeled with both sense and antisense probes. Localization was done with a lithium DMR microscope with Momarsky optics.
The paper reported that most of the difference in flower color among morning glories are due to how genes for pigment switch on or off, not mutations in the genes themselves. The scientists made this discovery using genes expression comparison and a statistic double in DN and DS ratio. The gene expression test evaluated a range of kind of activity being exhibited by the genes in different species, and the DNA and the DS ratio was about whenever or not the genes coding parts were withstanding natural selection. This ratio indicates the rate of change that occurs either due to amino acid substitution or due to silent change, allowing the author to assess whether the genus was involved naturally, undergone purified selection, or positive selection. The author found this pigment genus had different expressions levels between species, but the coding region of the genus are very conserved. The author used DN and DS tests that provide not stronger evidence of positive selection or strong evidence of purified selection, which indicates the genus structure are relatively stable. The author's main conclusion is that differences in flower colors were driven by regulation change in the gene's expression rather than change to the coding sequence of genes.
The study showed how the chloroplast DNA in the plant slowly changed over time. This was important because it helps scientists see and understand how plants have evolved and how their genes can be changed, yet some of them remain the same over time. The results were strong since they compared many plants and found patterns. At the end, they got the results they were hoping for because chloroplast genes are important for photosynthesis and normally stay the same. I also expected the same results, especially since the chloroplast and other important parts of the plant can't change. I think they could have studied more plants or studied more different parts of the DNA to see where it changed faster or slower.